Hello champions, I welcome you all to score 360 out of 360 in NEET Bio within 250 days. And today we are going to talk about the genetic code, and this is from the chapter molecular basis of inheritance. So I told you transcription is very much important. So along with transcription, all these things are very much important, like genetic code, translation, lack of Khorana and stuff like that, which will be coming along. I'll be showing you the type of questions as well, but I'm keeping you mentally prepared. And this topic is very simple. So why to lose your marks in simple things, right? So where is the cursor? Yeah, okay, cool. Now going ahead, what did we cover? We covered transcription of DNA, and if you have any doubts lingering around it, please feel very free to comment it down. I'll solve your doubts, or else you can watch my video what I have created for you guys. Yeah, and if you want the PDF of the same, join our WhatsApp channel. Now, the number of questions which was asked the previous year, it was 6. So, 6 answers, if you give it correctly, then you get 4 marks. So, 6 into 4 is 24 marks in total. But previous year, it was 10 questions from this chapter. So, 10 into 4, 40 marks from this chapter, and we cannot unsee this. So, we have to work hard in solving the questions of these chapter and covering all the topics, because this chapter is actually easy to score. It's not difficult. I don't know, people make it sound difficult, but it's not like that. So now let's see the question. Homework: what will be the sequence of mRNA produced by the following stretch of DNA? So they have given this DNA strand. In one they have written template strand, other one is coding strand. So I have to make that over here. If I suppose that this is I'll make in black. Okay. So this one basically is the template strand. Okay, this is in 3' to 5' direction, and there will be a coding strand over here. Okay, this will be in 5' to 3' direction. Okay, now new mRNA will be made on the template strand, on the basis of the template strand. Why? Because RNA polymerase can only make things from 5' to 3' end. Okay, so this will be the mRNA that will be formed, and how it will be formed. So over here, suppose it's A. So here it will be U instead of T, because you know, in the case of this thing, what do you call it? In the case of RNA, you do not have T, instead you have U, right? Similarly, in this mRNA, over here, if you suppose that this is T, then it will be A. If this is G, then it will be C. If it is C, it will be G. Okay, so whatever is over here, complementary to it, you get your mRNA. Now, you only think these two strands over here, okay, this strand and this strand were complementary to each other. So, A will be having T over here, T will be having A over here, G will be having C over here, C will be having G over here. Doesn't this and this seem similar to you? Except the fact that it has U and this has T, because in the case of DNA it will be having thymine, whereas in the case of RNA it has uracil. Rest everything is same. So can't we just say whatever is there in the coding strand, that is the exact thing that you get in the mRNA? So over here, rather than making all the codons, checking down all the codons, can't you just see whether the coding strand matches any of these things? Except you have to change the T into what? T into U. So everywhere where you have T, you have to make it U. So I am saying over here, this and this particular thing matches, this and this particular thing matches, but again I am talking about mRNA, but here it has given 3' end to 5' end, but my mRNA is formed in 5' to 3' end. So this is the correct answer, because this is given in 5' to 3' end direction only. Okay, listen, over here this and this is exactly same, except from the fact that T is being changed to U, it is replaced by U. Okay, this came in 2019, and I hope you were able to do that. Now, going ahead with today's topic. So, let's start this. Okay, who said codons should be in a group of 3? What are codons that you should be knowing first? So, basically, I told you the DNA basically forms the RNA. This is the mRNA, the messenger RNA. Okay, and mRNA basically will be having codes on it. For example, GUG, ACC, whatever is there. And now I say that we are going to read it in the format of 3, that is a triplet. Okay, and I am saying one more thing, that these 3 things together I call it a codon, and each codon is coding for an amino acid. That is what I say. How does codon code for amino acid? Basically, the storyline is that this particular mRNA basically get sandwiched between ribosome. Okay, so there is a one ribosomes unit and there will be another ribosomes unit over here. In between that you have got the mRNA sandwiched. Okay, now what will happen? There will be a tRNA coming which will be having an arm like this, and over here there will be an amino acid, specific amino acid. Okay, and there will be codon just opposite to it. For example, A is there, here there can be U, either C then G, here C then G. So basically this UGG containing tRNA will come in search of it, come and attach and see that okay, this is my exact complementary thing, and now I can give off my amino acid. Similarly, another thing will be coming in, another tRNA will be coming in with another amino acid and with another codon. Okay, so over here it's G, so it will be C, here it's U, so it will be A, here it's G, so it will be C. Okay, similarly all things comes and this amino acids they join together to form what, children? Proteins. And these things which are there on the tRNA, if I call these things as a codon, I call them as anticodon. What do I call it? Anticodon, the one that matches the codon. So anticodon, where do you find it? On the tRNA. Fine. This is how things work. Now, who said that codons should be in a group of 3? Who gave this information that codons basically will be in a group of 3? That is the question. So, it was George Gamow, children, a physicist, who argued that since there are only 4 bases, okay, since there are only 4 bases, that is adenine, guanine, cytosine and thymine, okay, they have to code, and all of them they have to code for 20 amino acids. How many? 20 amino acids. So, the code should constitute a combination of bases. So, can I say that A will be coding for amino acid, G can code for another amino acid, C can code for another amino acid and T for another amino acid? In that way, if I say, then it will be 1, 2, 3, 4 types of amino acid being formed. So, it cannot be taken individually. I cannot think that basically it's just individual amino acid which gives rise to an amino acid. Okay, sorry, individual what do you say, nitrogen bases which can give me an amino acid. That cannot be possible. In that case only 4 amino acid will be made, but we want how many, children? 20. Okay, so he suggested that in order to code for all the 20 amino acids, the code should be made up of 3 nucleotides. Okay, he said it should be made up of 3 nucleotides. So basically if I assume that it is made up of 2 nucleotides, still will I be able to make it? Will I be able to make 20 amino acids if I constitute 2 amino acids altogether? No. You have 4 things, okay, whatever possibilities is will come almost of 16, something like that, but we have 20 amino acids. So what do? Basically if 3 of them come together, probably we can make it code for an amino acid. It can be in different combination. For example, this 3 codes, A, G, C and then G, A, C and then CAG, whatever you do, you can have different combination ACG. All of these codons are going to give rise to amino acids, and in this way, how many amino acids, children? So 4 onto the power 3. How many I get? 64. So if it was, children, if it was just 2 nucleotides which were coming together, and if I had to make, if I had 4 of them, okay, 4 divided into 2, I get 16 amino acids. Whatever possibilities are there is just 16 possibilities. So 16 amino acids will be made. That's not my concern. I want how many, children? 20. So what do? Basically if I do this, if I have triplets, then what do? 4 raised to the power 3, I get 64 and it's all fine. I can include 20 amino acids or even more, right? In different combination. I get different codons. Okay? And these different codons, if they want, few of them can code for, okay, the different one can code for the same amino acid, making all of them together. If we calculate, for example, I am saying there are 60 of them, but 1, 2, 3 are coding for the same amino acid, that is alanine. So 3 of them are going for 1 amino acid, but still I can make 20 amino acid with pleasure, okay, and I have everything in my hand to do so. Okay, so 64 codons are possible. Don't think 64 amino acids are possible. It's not like that. We just have to make 20 amino acid. They can come in different combination. In how many different combination? 64 different combination. That is why I have said 64 codons. Now 1, 2, 3 codons can code for the same amino acid, that is alanine also. That is their choice. Okay, so it was George Gamow, who was what children, who was a physicist, who found this particular thing. And he suggested that it should be in the order of 3, then only things would work, otherwise it would not be working. What is the maximum possibility? 64 codons, please remember it. It's very much important. Don't go and write 64 amino acid, I'll kill you. Okay, George Gamow. Going ahead, Gobind Khorana and Marshall Nirenberg's contribution to genetic code. What did they do? So basically, the chemical method developed by Gobind Khorana was instrumental in synthesizing RNA molecules with defined combination of bases, homopolymers and copolymers. As in, what did this guy do? No, he made polymers. He, using his own brain, made homopolymers, as in all the bases, if you see, are same. Why do you call it homopolymer? Homo as in same, polymer as in when something is being made with the help of small things. So nucleotides, basically uridine nucleotides, are coming together to form a long chain. This is an RNA chain. Copolymer as in when you have A, G, C, G, C, T, sorry, not T, U, A, whatever is there. This is a copolymer where everything is there. So over here, homopolymer was made, okay, with the help of Gobind Khorana. He was the one who thought that, okay, we can make this homopolymer and copolymer as well. But what is the use of doing this? That is the next question. So after that, one more information is given over here, that is Severo-Ochoa enzyme, that is polynucleotide phosphorylase. Polynucleotide, so many nucleotides. Phosphorylase was helpful in polymerizing RNA with defined sequences in template independent manner, enzymatic synthesis of RNA. So basically it's saying it was made. This particular thing was made, but with the help of what, children? They have mentioned the name of the enzyme. What was the name of the enzyme? Severo-Ochoa enzyme. Severo-Ochoa enzyme. You also call it polynucleotide, so many nucleotides, okay, polynucleotide phosphorylase. You also call it polynucleotide phosphorylase. So this particular enzyme was used for making the bonds like this, or else obviously we cannot make the bonds. Okay, it is DNA independent. So you are making an RNA without the help of any templates. Usually what happens, your DNA is there, something is copied, mRNA is being formed over here, right? There is a template strand, but here without the help of template strand, things were made, and that is why we needed an enzyme, Severo-Ochoa enzyme, that is polynucleotide phosphorylase was used. Okay. Now, Marshall Nirenberg's cell-free system for protein synthesis finally had the code to be deciphered. That means, basically what did he do? Next person who is in there, Marshall Nirenberg. So, I am writing Marshall Nirenberg, okay, there is a double L. What did he do was, he tried to synthesize proteins out of this particular homopolymer, and he found that it was phenylalanine being formed. And phenylalanine is being what children? Phenylalanine is an amino acid, children. Is what? Is an amino acid. So amino acids, basically when they come together, what do they make? They make the proteins, right? So basically over here he found, Marshall Nirenberg, he found that so many phenylalanine was being formed. Okay, nothing else, just phenylalanine amino acid was being formed. And with this, he understood one thing, that probably UUU is coding for this amino acid, that is phenylalanine. Similarly, next UUU was also coding for phenylalanine. That is why it was all amino acids, that phenylalanine amino acid, that was being formed. Did you get it? I'll tell once more. Gobind Khorana, he, with the help of the enzymes that are present over here, that is Severo-Ochoa enzyme, that is polynucleotide phosphorylase. So without the help of a template strand, he made the RNA. One he made, that was homopolymer, the other one was copolymer. Homopolymer as in all the nucleotides are same. Copolymer when they are different. Okay, so basically this homopolymer that he made, okay, was tested on by Marshall Nirenberg, and what he found was astonishing. He found that it was just a long strand of phenylalanine that was being formed. Probably because this is a homopolymer and all of them, they make a codon, and UUU, okay, UUU probably stands for phenylalanine. alanine, which is why it is being made. So he got to know that each codon is going to give rise to each amino acid. So when he did this, probably, then he got to know different amino acids are being made, because obviously there are different codes as well. You get it? So this was done by them. Please remember her, Gobind, Khorana and them. Okay? And before that you have George Gamow, who was a physicist. Fine. Now, genetic code was made. After that, there were certain salient features which were also found in the genetic code. What was it? Let's study. The codon is triplet. You know that. The codon is what, children? In the form of triplet. And 61 codons code for amino acids, and 3 codons do not code for any amino acid. Hence, they function as stop codons. For example, if mRNA is there, tRNAs are coming, binding over here, and amino acids are also coming. So similarly, you know that there are so many amino acid chain being formed. Okay. Now we do not want it have reached the end. Over here there will be a codon which does not code for any protein. Okay. And such codons are 3 in number. Such codons basically are 3 in number. 3 different combination of codons are there, which says that you have to stop. I'll tell you what is the name of it. It's UAA, UAG, UGA. Okay, UAA, UAG, UGA. These are the 3 codons which do not code for any proteins. So, not code for any amino acids. So, I call them stop codons. So, how many codons about which we learned was 64. Out of that, 63 are coding, good children. They are, not 63, 61 are good ones. They code for amino acids. And 3 of them basically are known as stop codons. They are not going to code for anything, children. Okay. Now, what is the name? Please remember it. UGA, UAG, UA. I am not able to remember the formula for remembering it. Uga, Uag, Ua. Okay. Either way, remember it. You get away. You always go. Something like that. You make your own thing, whatever is comfortable for you. But it's UGA, UAG and UAA. Okay. Now going ahead, some amino acids are coded by more than one codon, hence the code is degenerate. So basically, one amino acid, for example, I say phenylalanine or I say leucine. Leucine is an amino acid. Okay. I say leucine is being formed by this codon XXX and then this codon XXX. And then another codon XXX. So, there are different codons coding for one amino acid. Hence, what is the feature? Since amino acids are coded by more than one codon, hence the code is degenerate. What do you call this feature as? Degenerate, when more than one codon is coming to form what? The amino acid. Okay. Now, the codon is read in mRNA in a contiguous fashion. What do you mean by contiguous fashion? Basically, it's continuous. Okay, there is no punctuations in between. So basically, I can say that when a codon is read A, U, G, C, G, A, over here there is no full stop, there is no comma, nothing. Over here it is read like this 1, and this is 2. And it goes on. So there is no comma, there is no punctuation, it is known as contiguous. Okay, it is known as contiguous. So codons are degenerate, children. Why? Because there are multiple codons coding for the same amino acid. Okay, so next thing that we studied is contiguous, there is no punctuation in between. Now the code is nearly universal. Okay, it will be nearly universal, it's not said completely universal, nearly universal as in basically all different organisms. Okay, in them if the mRNA is having this AUG will always code for methionine and nothing else. Whether it be any organism, it will be coding for just methionine. If this is a codon, then this codon codes for one particular amino acid in general, nothing else. This is nearly universal. Okay. So, for example, from bacteria to human, UUU would always code for phenylalanine. Okay. So, if it's bacteria, if it's human, for everyone, UUU means basically phenylalanine. Now, some exceptions to this rule have been found in mitochondrial codons and some protozoans. So, exceptions are also there which you need to remember. Okay. What is it, children? Mitochondrial codons and in some protozoans. Fine. Will you be able to remember it? Where is it? found as an exception mitochondrial codon and in some protozoans. So mitochondria also have codons, so them and the protozoans. Now AUG has dual functions. When I talk about AUG, it is said that it has dual function. One is that it codes for methionine. So methionine is coded by only one codon. It's not like leucine. It's just coded by one codon, and it is AUG. Now AUG has dual function, and the dual function is that whenever you find AUG in an amino acid, in a mRNA strand, that means from this particular position the translation will start. So it is also acting as a start codon or the initiator codon. Okay, what do you call this? You call this codon as initiator codon because it is going to initiate things. Okay, so AUG has dual function. What is that? One function is to form methionine, to code for methionine, and the other one is to initiate the process of translation. Okay, now UAA, UAG, UGA are stop terminator codons. That is also given. So what did we understand, children? We have 64 codons, out of which 61 codes for amino acid and 3 codons basically are known as stop codons. Now next thing, some amino acids are being coded by more than one codon that you can see over here. So what do you call this as? You call them as degenerate. Okay, you call this is very degrading. Okay, these are basically, so leucine thinks that I am only coded by one amino acid, one codon, but it's not like that. Okay, or probably this guy is thinking that I am only coding for leucine, but then comes to know that this is also coding, this is also coding. So it's very degenerate thing that is happening over here. You can think like that, whatever you, I know it's too much of drama from my end, but yes. Okay, now the codon is read. Read in mRNA in a contiguous fashion. Obviously there are no punctuations, and the code is nearly universal except mitochondrial codon and the protozoan. Okay, protozoans comes under Protista. So there is a kingdom Protista, and in Protista comes the protozoan. So amoeba, paramecium, everything, and comes in this category. Okay. Now AUG has dual function. One function is to code for methionine, which is an amino acid, and the second function is to act as an initiator codon. It initiates the process of translation. Then you have UAA, UAG, UGA, which are the terminator codons. Cool? Works? Now going ahead, we read about it. This is the chart that is being made in your book for the genetic code. Ma'am, do we have to remember it? Yes, children, you have to remember it. The name of the codons, the name of everything. It's very easy. So what do? Okay, I make that chart over here. I'll tell you how to make things. Okay, so we need to make 4 boxes. 1, 2, 3 and 4. Similarly over here also. 1, 2, 3 and 4. Cool? Over here we'll put letters. UCAG. In this format only write. UCAG. Okay? Now here you'll write everything. UCAG, 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 UCAG. Cool? Now, codons goes like this. U, and there U. Okay, so UCAG I can write over here, and then I can copy everything from here only. Okay, so it was U there. Now U, C. Now it will be U, A. There it will be U, G. Okay, you have to write like this. Got it? It's easy, no? It's easy, or is it difficult? Or did I mess it up a bit? I messed it up. It's U. Sorry, my dear champions. Okay, so U, C, A, and G. Now it works. Okay, C, A, G. Okay. Now next one. How will it be? It will be C, A, C-A-G. Okay, ayyo. Just a second. Why is your teacher doing like this? Okay. U-C-A-G, just like that I copied over here. Okay. Now C-U will be there. So C-U, C-U, C-U will be the thing. Now if I want to make it over here, this one. Okay. So it will be A, C, and then what? U-C-A-G. So again it will be A, C, A, C, A, C. So it's ACU, ACC, ACA, ACG. Fine. Now if I want to make it over here, so it will be C, it will be A, and then it will be U. Over here I am writing UCAG. Similarly, this will be CA, CA, CA. Okay. Now if I want to write it over here, so it will be G, G, and then U, C, A, G. So it will be G, G, C, G, G, A, G, G, G. Okay. So in this format you will get 64 children. How many of them you will get? 64. Okay. And you have to remember the names. So I'll tell you a trick to remember the names, okay, of this. PHE stands for phenylalanine, okay. What does it stand for? Phenylalanine. LEU stands for leucine, okay. So I'm writing it over here for you to understand, then I'll give you the trick. So PHE stands for phenylalanine, okay. LEU stands for leucine. LEU goes up till here, and then you have isoleucine, ILE, okay. What is it? Isoleucine. This is your methionine. Methionine has just one codon if you're seeing it, okay. Now this is with your valine. So phenylalanine, leucine, isoleucine, methionine, valine, okay. Now we have a trick. Pirates love isolated mysterious valley. Pirates, they love isolated mysterious valley, where P stands for what, children? Phenylalanine. L stands for leucine, I stands for isoleucine, M stands for methionine, and V stands for valine. So, pirates love isolated mysterious valleys. Okay? Will you be able to remember it? Now you have to see how many codons actually code. So for phenylalanine you have 2, for leucine you have 2. So this box is shared by 2 people, phenylalanine and leucine. Then you have full leucine, then you have isoleucine, but then one guy over here is methionine, AUG. Okay? Then comes whole valine. Now second thing, it's very simple. Okay? Second one is very simple. One whole box is for serine. One box is for serine. The other box is for proline. The other box is for threonine. The other box is for alanine. Okay? So we have one box full for serine, and then proline, and then threonine, and then alanine over here. So smart people take actions. S stands for what, children? Serine. P stands for what, children? Tell me. P stands for what? Proline. Okay, everyone ends in Ne, okay? T stands for threonine, and A stands for alanine. Okay, SPTA. Serine, proline, threonine, alanine. Please do not get confused between threonine and tyrosine. It's different. Okay, tyrosine comes later. Over here you have to remember threonine. So here the boxes are kind of same for all. Over here phenylalanine and leucine were sharing a box, then it was full leucine, then isoleucine was there, but one was given to methionine and then valine. Now the next one is tyrosine. Now everything here in this particular line is coming in 2 format. Okay, 2 for tyrosine, 2 stop codons, 2 histidine, 2 glutamine, 2 aspartame, 2 lysine, and then 2 aspargine. Sorry, this is aspargine, this is aspartate, and this is glutamate. Not able to remember? I will make you remember. Okay, so 2 is there. Just remember that. Okay. Now over here, tyrosine. T for tyrosine. 2 tyrosine are there. 2 stop codons are there. For H, it is histidine. So 2 histidine is there. For G, it is glutamine. Okay, so glutamine is different from glutamate. So glutamine is there, and aspargine is there. Aspargine, glutamine, aspargine is there. L stands for lysine. We covered leucine, not lysine. Okay, this is lysine. A stands for aspartate, and G stands for glutamate. So aspartic acid and glutamic acid comes to the end. So this is 2, 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 this is 2. Okay. tyrosine, stop 2, then histidine, then glutamine, then asparagine, then lysine, then aspartate and glutamate. So tree spread happiness growing apple, lemon and grapes. Okay, if it helps you can think like that, but listen, without no help also you can study. If you think, if you just sit and look at it for some time also, it will be fitted in your brain automatically. Okay, now coming to this, they have alternate things that have similar boxes and alternate where the things are very different. So in this particular box, in whole this box, it is arginine. It is what, children? Arginine. And this whole box, it is glycine. Okay, one whole box is arginine, one whole box is glycine. And over here you have cysteine. There is one stop codon. There is one for tryptophan. So tryptophan is also like methionine, it also has just one codon. So I am writing it over here. Methionine and tryptophan has just one codon, children. Okay, they have just one codon. So cysteine, stop, tryptophan, then arginine full, then glycine full. Over here you have serine. Again, so serine was over here, 4 of them was there, but still it is not fitted up. So serine is there, then you have arginine. So you can remember like this, arginine had a separate thing, but still it needed some space. Similarly, serine also had a separate thing, but still it needed some space. Okay, so you have cysteine, stop, then you have tryptophan, then you have arginine, then serine, then arginine again, and then glycine. Okay, so over here, C stands for cysteine, S stands for stop, there are 2 cysteine, okay. Then one tryptophan is there, only one tryptophan is there, this is just one. A for arginine, so whole box is for arginine, they are 4 in number. Then S is for again serine, which was over here. A is again for arginine, so basically 2 here, 2 here. And then G stands for glycine. For glycine also it has one whole box. So I know it's not like very easy to remember it one go, but I am telling you, trust me on this thing. If you sit and go through certain things, I don't think that you need this also. Okay, just by looking at this chart, you will be able to understand a lot of things. I'm pretty sure about that. Okay, but just you need to know how to make the chart. That is what I have written over here. UCAG, UCAG. There you have to look at that and then you have to write it. Okay, in every box there will be 4, one coming ending in UCAG. All of them will be ending in UCAG. Works? Okay, what is the name of the start codon? It is AUG. What is the name of stop codon? It is UAA, UAG, UGA. These are the stop codons. This is the start codon. Okay. So if I am saying that you have 20 amino acids, how many codons are there, children? If there are 20 amino acids, how many codons? 20 codons? 20 codons. I am not talking about the nitrogen bases. I am saying about the codons. 1, 2, 3 come together to become one codon, and one codon gives rise to one amino acid. So 20 amino acids means, that means how many codons, children? 20 codons. It will be what? 20 codons. Okay. The questions can come like this. You have to make sure that you know the answers as well. Now, over here, let's start. So, starting with this question. If the distance between 2 consecutive base pair is 0.34 nm and the total number of base pair of a DNA double helix in a typical mammalian cell is 6.6 into 10 to the power 9 BP, then the length of the DNA is approximately equal to what? They are saying if the distance between 2 base pairs, okay, there is one base pair over here, there is another base pair over here, the distance is 0.34 nm, okay, and they are saying the number of base pairs are 6.6 into 10 to the power 9, this is of eukaryotes. What is the length of DNA? Can you guess it? I'll give you 1 minute time for guessing it. Pause the video and try to guess it and then tell me the answer. What is the length of the DNA? Basically, when I do into, what do I get, children? It's 2.2 meters. It will be 2.2 meters because when I calculate nm into meters, it comes to be 10 to power minus 9. Okay, then when I calculate it, I get 2.2 meters. Works? Okay, going ahead. So, what is the answer for this thing? This is the answer. Okay. Now, Under which of the following conditions there will be no change in the reading frame of the following mRNA? Yeah, with this I want to tell you one more thing. Okay, so listen to me carefully again. When okay, this is a codon, this is a codon, this is a codon. From here, if I am deleting one, okay, there is frame shift mutation, as in the frame was initially this, but now since I have deleted this, what will happen? This will be the 3 codon, this will be the another 3 codon. So the whole codon has changed. That means the amino acid which was being formed, that is also changing. So when you do insertion or deletion or something, when you insert something, insertion or deletion, when you do it, basically codons are changed. Okay, basically the codons which are coding for the amino acid, it will be changed. This is the case when I insert or delete either 1 or 2. What if I delete the whole 3 things together? Is this changing the frame? No. Okay, initially if it was like this, okay, if I initially this was one, this was another, and this was the third one, I remove the 3 things together. Okay, I remove the 3 things together. Is it causing any difference in my frame there? This will still code for that amino acid. That one particular amino acid which was over there, that would be missing, but this will again code for the amino acid. So please remember, when 3 codons are removed, 3 nitrogen bases are removed, it does not cause frame shift mutations. When 3 amino acids are removed, it does not cause frame shift mutations. Okay. Now, under which of the following conditions there will be no change in the reading frame of the following mRNA? This is the thing. Okay. Deletion of GGU from 7th, 8th and 9th position, insertion of G at 5th position, deletion of G from 5th position, insertion of A and G at 4th and 5th. Now, they are asking you, there should not be any change in the reading frame. For that to happen, you have to remove 3 codons together. If you remove these 3 codons together, then possibly things won't be changing, because this is one codon, this is another codon, this is another codon, this is another codon, right? So when 3 of them are gone, okay, when 3 of them are gone, this is not going to change the frame of the things. Okay, even though amino acids won't be formed, that particular amino acid is not formed, but the frame is not changed. Mutation will happen for sure, but that frame is not changed. You have to understand that. So the answer will be this. Okay, this came in NEET 2019. Going ahead, if there are 999 bases in an RNA that code for a protein with 333 amino acids. So, check that 333 amino acids are there, that means 333 codons are there. Agree? Now, and the base at 901 is deleted. So, there are how many? 999 ones, nitrogen bases, and the one that is in 901 is deleted. That nitrogen base is deleted, such that the length of the RNA becomes 998. Now it has become 998. You are asked how many codons will be altered. So before 901, everything was still making this same thing. Everything was still making, the codons were still forming the things over there. From here to here, the things have changed. From here to here, the things have changed. If I minus this thing, 999 minus 901, I'll get 98 something. Okay, basically 98 amino acids or 98 nitrogen bases are there. 98 what, children? 98 nitrogen bases are there if they come in the format of 3. So divided by 3 if I do, I get something like 32.3 or something like that, basically 33. So 33 codons will be affected. Why did I divide with 3? What is this calculation? Let me tell you. So 901, that this position I am changing, I am removing one thing. So from here to 999, whole everything will be changed. Okay, so if I minus it, almost I get 98. 98 nitrogen bases are there. Nitrogen bases, they come in the form of triplets. So 98 of them, if I divide by 3, I will get to know the codons. So it is almost a 132.3. Basically I should be doing 99. Similarly, yeah, it's basically this thing only. Okay, so I get what? 32.3. If I divide 98 nitrogen bases divide by 3 because I want to find the number of codons, I get this number which is almost close to this. So this is the answer. It's 33. It's 33 codon which will be altered. So you have to know that thing. Simple. You minus the whatever content is there and then you divide it by 3 because these are the nitrogen bases. So you have to divide them by 3, then only you will get to know the number of codons because codons come in the form of triplets. Right? So that is the answer, children. Okay? Going ahead, which one of the following is a starter codon or start codon or initiator codon? You tell me. It's AUG, which also codes for methionine. Cool. Now, whose experiments cracked the DNA and discovered unequivocally that a genetic code is triplet? Who was that particular person? It was Nirenberg and Mathai. Basically, Mathai is not given, but he was also the one who got the Nobel Prize for finding this. So it is Marshall Nirenberg and Mathai who were responsible for this. Okay, who said that okay, this is the thing. Triplet is pakka there, and this triplet codons are responsible for each amino acids as well. Okay, so this is the correct answer. This came in 2009. Now, so the whole questions, if we see, the answer for this was 2.2 meters. The answer was deletion of GGU won't be changing because 3 codons does not make any change. Here, the number of altered codons will be 33. Which one of the following is a starter codon, children? You know it is AUG, that is methionine. And whose experiments cracked the discovery of triplet codons? It is Nirenberg and Mathai. Cool. Now, one question that I have to give it for you is this. What would happen if in a gene encoding a polypeptide of 50 amino acid, the 25th codon is mutated to UAA? So, from here, it is now mutated to UAA. What will be the change? Okay, that's the only thing that you need to find. You can put down the comments, that is what is the answer for this particular homework question. Okay. And that's all. If you think that this particular video was helpful for you, then do not forget to subscribe to the channel, and I wish you have a good day. Bye bye. Take care.